guys, welcome to our newest video. Today we're going to be talking to you guys about DNA. We're going to first ask a couple of people a question. What is DNA?
I have no idea what the heck DNA is.
Okay, thank
Great.
you, sir. I have no clue. Thank you. Great money going towards that.
You know, clearly there's a misunderstanding here. We've got to pass on to people what DNA actually is. All right, well here we can start with where can you find DNA? Well, pretty much you can find DNA almost everywhere. You can find DNA in, say, a 1, 2, 3, 4, four-legged dog. Next, you can find even DNA in a long snake.
You can even find DNA in our friend Hal. Now, you might be asking, where in these creatures is DNA found? Well, first we need to take a step back. If you zoom in very close, you might see some cells. And inside these cells, in the nucleus, is DNA. And DNA makes up the genetic code that makes each cell unique.
Well, looking at what the DNA structure actually is, when you look at it, it looks like it's pretty much just a twisted ladder thingy. Well, that twisted ladder thingy is actually called a double helix.
Yeah, so a double helix, the sides of the ladder, are made up from a phosphate, a sugar, and a base pair. And these base pairs are adenine, guanine, thymine, and cytosine.
Now, all these base pairs have to go together with one another. So adenine goes with thymine, and guanine goes with cytosine. How do these base pairs go together? Well, they're connected by a hydrogen bond right in the middle of the two. All right, well, you may be asking, how do these base pairs really connect with one another to form this double helix? Well, it's actually pretty simple. Right here in the center of the double helix, you see a hydrogen bond.
Now, these hydrogen bonds are here because they're easy to break, which allows easy access to replication, which we're now going to dive deeper into. Another vital part about how the nitrogenous bases pair together is that some are purines and some are pyrimidines.
Well, let's first start with the basics about what are pyrimidines and what are purines and why do they go together. Well, pyrimidines are a bit bigger than purines. Just like a jigsaw puzzle, when you have a big jigsaw piece, you need a smaller piece to connect those two pieces together. That's exactly the same when you have the base pairs here. When looking at the two purines, you have A, adenine, and G, guanine. Those pair with the two pyrimidines, which are T, thymine, and C, cytosine. Like to learn how DNA actually replicates?
Okay, well first, an enzyme called polymerase comes and unzips the double helix. And then in swims a new daughter strand to pair with the parent strand. So now I have two completely new DNAs.
In our original old DNA strand, it is made up of two parent strands, showed in the gray color on both sides of the double helix.
After replication, there becomes two identical strands, one with a daughter and one with the parent.
Alright, so you may be asking, why do we need DNA replication and why is it even important?
Well, we can compare DNA replication to, say, a baby.
It's a pretty big baby.
It is a pretty big baby. See, if there was no new babies in the world, eventually the human race would be extinct. This is similar to DNA strands because if new DNA wasn't produced, there wouldn't be any new way for cells to get the genetic code they need. Where does DNA replication occur?
DNA replication occurs in the nucleus of the eukaryotic cell. Alright, Jack, now let's shift gears real quickly. Let's shift from DNA to RNA. Well, what exactly is RNA? RNA is a ribonucleic acid. This nucleic acid is present in all living things, just like DNA is. Its principal role is to act as a messenger, carrying instructions from DNA for controlling and synthesizing proteins. Alright, well, technically RNA and DNA do work together, but that doesn't mean they're necessarily the same. In many ways, they're actually pretty different.
DNA is a long polymer with deoxyriboses and a phosphate backbone, having 4 different nitrogenous bases: adenine, guanine, cytosine, and thymine. RNA is a polymer with a ribose and backbone phosphate, 4 different nitrogenous bases: adenine, guanine, thymine, urine.
Transcription is the first step of gene expression, in which a particular segment of DNA is copied into an RNA by the enzyme RNA polymerase.
Now, enzymes are very vital in the role of tRNA.
Exactly right, Jack. Without enzymes, there would be no such thing as tRNA. More specifically, the enzymes within tRNA help them reattach and detach from their existing strands. All right, now, Jack, I just have a simple question for you. I'm talking about transcription. We're learning all this stuff about transcription, what it is, all this sort of things. But really, why is it all that important?
Well, you see, transcription is important because it's what brings the code from the nucleus all the way over to the...
A ribosome?
Oh, yes, sir, ribosome. And see, once it gets to the ribosome, it tells the ribosome what proteins it needs to make the cell.
All right, so when talking about the direction of transcription, we're going to know that it's going to go from 3' all the way to 5'. In transcription, the RNA polymerase is going to move from 3' all the way over to 5'. Once we get to 5', that means that mRNA is going to be produced, and it's going to go back from 5' all the way back over to 3'. That's going to be your basic knowledge of knowing the direction of transcription. So, all right, guys, unlike transcription, where everything happens in the nucleus,
Translation happens in the cytoplasm.
All right, good afternoon, everyone. I'm sitting up here on this desk or table to talk to you guys and read you guys a short story about what the genetic code actually is. Bear with me for the next couple of minutes, please. The genetic code is a set of rules by which information is encoded within genetic material. This is either by the DNA or the mRNA. It is translated into proteins by all living cells. Biological decoding is accomplished by the ribosome, which links amino acids in order specified by mRNA using transfer RNA molecules to carry amino acids and read the mRNA 3 nucleotides at a time. Thank you, guys. All right, so when talking about translation, the role of the proteins and the structure of them go hand in hand with one another. The structure of the proteins in translation is made up of amino acid chains. And what these amino acid chains do, and what more specifically the proteins do, is they help the whole body function. All right, so what the enzymes do in translation is that they attach one end of the amino acid chain from the tRNA strand to a different amino acid chain on another tRNA strand. All right, guys, so now we're going to move into some of the advanced stuff. If you guys need me to slow down, too bad. I'm going to be talking to you guys today about the 3 different types of RNA. I did earlier, but this time I'm going to get into more detail about all of them. What makes them all different from one another is that each RNA is encoded by its own type of gene. For instance, mRNA, which is messenger RNA, encodes the amino acid sequence of a polypeptide. tRNA, which is the transfer RNA, brings amino acids to ribosomes during translation. And lastly, in rRNA, which is the ribosomal RNA with ribosomal proteins, makes up the ribosomes, the organelles that translate the mRNA. Alright, thanks guys for watching our video. If you guys want to subscribe, please do. Just kidding, we don't have any subscriptions.
Also, if you want to see another video, click on this annotation. Oh, I'm just kidding. I don't know how to make annotations, and we also do not have more videos.
Thank you guys.